So between the two layers, the membrane is called the perimitochondrial space, and the cristae along with the inner membrane regularly spaced stopped particles are known as the oxysomes or F1 particles. So the matrix contains the proteins, lipids, and oxidative enzymes also there. The mitochondria are the site of the cellular respiration involved in the oxidation of food and synthesis of ATP. Therefore, the mitochondria is also called as a powerhouse of the cell. So another one is the plastids. The term plastids was coined by the Schimper. They occur commonly in eukaryotic plant and they may be colorless or colored. So chloroplast, these chloroplasts also here. So this is the chloroplast here. The chloroplasts are usually ellipsoidal or discoid in shape. They measure around 5 to 10 millimicrons in long and 2 to 3 millimicrons thick. There are also two membranes are surrounded by these chloroplasts. The outer membrane generally appears smooth, where the inner membrane is covered with the finger-like the projections into the stroma. The matrix, which is also called as stroma, consists of connecting solutions of proteins. The membrane inside the chloroplast form the secondary thin sac called lamellae. The stroma contains ribosomes and circular naked DNA, lipid globules, starch granules also there. So these units are also known as the quantosomes. Each quantosome consists of around 300 to 600 chloroplast, carotenoids, and some proteins, enzymes also there. What is the function of the chloroplast? Here, there are the mainly phosphate in the photosynthesis. So that's why to prepare the carbohydrate. And also colorless chloroplastids help in the store the food material. So which is the leucoplast, they are to stored in the uh, the starch in amyloplast and lipids are elaplast and proteinoplast, they are reserved in the proteins. So chromoplast gives the colors to the flowers and fruits also. So here the another one, some micro bodies also present inside the cell. The microbodies are two types are there, peroxisomes and glycosomes are there. So peroxisomes, so there are the along with the some chloroplast in the chloromesophils of C3 plants. And glycosomes, there are the rich in endosperm cells. They possess the enzymes for the converting the fats into carbohydrates. So some lysosomes also present in the cell. So these are the single membrane bounded organ. These lysosomes contain a variety of 10 to 12 types of the hydrolytic enzymes are there. So these lysozymes, so they are called as the suicidal bags of the cells. Means during the cell death, these enzymes are revealed and outside and result in the dead cells of there. In this way, helps in the autolysis of the cell. So that's why we call as the suicidal bath of the cell. Microtubules and microfibrils also there, microfilaments also present. Some vesicles, vacuoles also there in the cytoplasm of the plant cell and as well as animal cells also. So here these vacuoles are bounded a single semi-permeable membrane that is known as a tonoplast. These vacuoles are filled with the fluid like material which is called as a cell sap. So structure function of the cell membrane. So these inner the cell wall and surrounded the cytoplasm is a thin and delicate and flexible cell membrane is present, which is called as a plasma membrane or plasma lemma. So which is made up of the some lipids, proteins are there. So ultrastructure of the plasma membrane. The several biologists have proposed the different theories also there to structure of the plasma membrane. There are the two types are there, bilayer model and micellar model. Here bilayer model are lipid bilayer model, also sandwich model, and here Daniel Davidson model, fluid mosaic model. Different types of models are there. Here we explain the fluid mosaic model. So fluid mosaic model is proposed by the Singer and Nicholson. So according to this theory, the proteins are depressed within the bimolecular lipid layer in the form of the mosaic type. This is just like a protein iceberg floating in the lipid sea. So in these proteins are arranged into different types. Some are the present in the surface of the membrane and other there are referred as the extinct proteins, and some proteins are present embedded in the lipid layer. They are referred as the instinct proteins are there. So this fluid muscle model is in the dynamic structure with the permeability barrier are there also serve in the solvent from integral proteins and lipids also there. Lipids and integral proteins in the biomolecular layer are free to move laterally within the liquid solvent. Okay, so this the carbohydrate molecules of the glycoproteins and glycolipids are there in the this membrane. So they also have critical role in the phenomena of cell to cell and recognize the interaction. Okay. Another one is the nucleus. What is the nucleus here? This is nucleus is there. So the nucleus is consist of the following major parts are there. That is the nuclear membrane, nuclear sap, chromatin fibers, and nucleolus. So what is the nuclear membrane? This nucleus is surrounded by the envelope that is there. That one two membranes are there. That one we call as a nuclear membrane. So between these nuclear membrane, the space is there. That space we call as the perinuclear space. So nuclear sap is the interior of the nucleus is filled with the transparent and granules and acidophilic fluid called as the nucleoplasm, which is contain the RNA and hydrolytic enzymes. So RB. So here Some chromatin fibers also there. These chromatin fibers here elongated, some thread-like structures are there that is suspended throughout the nucleoplasm called as a chromatin fibers. So these chromatin fibers are there. So which is the chemically DNA and proteins are there. So nucleus consists of a spherical, prominent ball-like body, which is known as the nucleolus. Which is generally only one nucleus is present in each nucleus. Okay. So here the nuclear morphology. So based on the nuclear morphology here, generally only one nucleus is present in cell, but sometimes apetal cell or latex have many nucleus also present. The shape is depending on the cell, which may be spherical and spheroidal types is there. So number of the chromosomes here, the chromatin substance nucleus forms chromosome during the cell division stage. So if the one set of chromosome is present, which is known as a haploid condition, if two sets of chromosome, which is called as a diploid condition. So this is the function of the chromosomes. Here, function of the nucleus. Nucleus undergoes division of during the cell division. Throughout the mitosis division, it is helps to undergoes to the growth and development. Where the meiotic division, it is helps to the formation of the gametes, which is required for the sexual reproduction. And it plays an important role in the heredity as it contains the DNA, it is in the chromosomes. The nucleolus present in the nucleus play an important role in the synthesis of ribosomes. And nuclear cells have DNA replication and RNA transcription take place. So these nucleus control the all the metabolic activity of the plant cells. This is the nucleus. So and another third unit is the that is the this, and nucleic acids. So this chemical construct of the nucleic acid here there are the three types of the substances are there, that is the phosphoric acid, pentose phosphate, and the nitrogen bases are there. So phosphoric acid here, this phosphoric acid has the three reactive hydrolysis group. There are the involved in the formation of sugar phosphate. There are the backbone of the DNA. This is the phosphoric acid and glucose pentose sugar means here there are the two types of the sugars are present in the nucleic acid. The pentose sugar present in the RNA, they are called the deoxyribose, where the nucleic acid gets name, but the DNA consists of deoxyribose. Okay. Nitrogen bases are there. So here these bases they are called these nitrogenous bases are here two types are there, that is the purines and pyrimidines. So purines are two rings structure. One contains a six membrane pyrimidine ring of the benzene and five membrane imidazole ring are there. So here purines are two types are there, adenine and guanine. So both are found in the RNA and DNA is also there. So pyrimidines here, pyrimidines are consist of the six membrane pyrimidine ring. There are the here pyrimidines are three types are there. What is the cytosine, thymine, and uracil? In DNA, thymine and cytosine are present, whereas the RNA instead of the thymine, uracil is present. That is the pyrimidines are there. So nucleosides. So nucleosides means the nitrogen base linked to pentose sugar are known as a nucleoside. So what are the nucleotides here? A nucleoside is formed when the phosphate group is attached to the pi carbon of the pentose sugar, which is called as a nucleoside. And these nucleosides produced by the different ribosomes of RNA are adenine acid and 5-granate acid. These are the there. So structure of the DNA. So here the DNA molecule is double stranded, which is the two polynucleotide strands are coiled like together to form the DNA double helix. So each of these two strands of the DNA molecule is the polymer of the deoxyribonucleic acid, and they are known as the polynucleotide. At the uh, here at the end of the polynucleotide chain, here 5 carbon pentose has 3 of the OH, while the 3 is the carbon of the pentose of the opposite, it is end free of the OH. So here they are explain the DNA structure here Watson and Crick model. So James Watson and Crick proposed the structure of the DNA on the basis of the chemical and physical and biological properties of the DNA. So which is the DNA molecule is made up of the two polynucleotides. Hmm. So here the two strands are coiled together forming the DNA double helix. The two strands of DNA molecule are oriented anti-parallel to each other. So that's why each polynucleotide is composed of the many nucleotides jointly and phosphodiester linked between their sugar and phosphate residues. So these two are the backbone of the DNA. The nitrogen base is organized in the stepwise manner. They are parallel to each other, connecting the both strands of the DNA. Here, so here there are the adenine is always bound with the thymine. The guanine pair which is the cytosine. So adenine and thymine they have the two hydrogen bonds, whereas the guanine and cytosine has three hydrogen bonds are there. So these two strands of DNA molecule are coiled together in the right hand helix, forming the DNA double helix. So the diameter of these helix in 220 angstroms, where the pitch of is 34 angstroms. Based on the arrangement of the nitrogen bases in one strand, the sequence of bases other strand is determined. So here the genetic formation of the present each DNA strand is known as the template strand. Replication of the DNA. Here, the process of the which is DNA molecule produce the identical copy is described as a DNA replication. So the new formed DNA resemble to the parental strand in all aspects. That is there are three steps are there here. Distributive replication and conservative replication and semi-conservative replication. So differential replication, the fragment of DNA gets separate randomly and new daughter DNA is synthesized. In this, some segments of the DNA contain the parental DNA and some newly synthesized DNA is present. Conservative replication, the parental DNA undergoes to duplication resulting two daughter DNA molecule. And conservative replication. Here, two strands of DNA molecule uncoil and each strand produce the daughter complementary strand. It is the result in the formation of the two daughter DNA molecules. So here, the semi-conservative model of the DNA replication. In this, the double-strand DNA unwinded and produced two single DNA fragments. Each single strand acts as a template strand for the production of the new daughter DNA. This model is termed as a semi-conservative DNA replication. Here, the mechanism of semi-conservative DNA replication. Here, the DNA replication includes three major steps are there. That is the initiation and elongation and termination. Here, three steps are involved in the DNA replication. Here, the initiation DNA replication begins at a certain unique and fixed the point called as a origin. So the initiation begins with the uncoiled of double DNA strand. Two enzymes
డబ్బా సోమరాజు ఎక్కడ ఉంది?
and the DNA helicase binding to origin point and
సోమరాజు సోమరాజు
unwinding
ఈరోజు రెస్టా?
and separation of the
ఇది బి షిఫ్ట్.
complementary strands of the DNA double helix.
ఈరోజు నైట్ షిఫ్ట్ ఏంది? ఈ మాట్లాడుతానండి. ఎక్కడ ఇయ్యాలి మీకు డబ్బా?
Okay. So elongation, the DNA catalyzed by the enzyme DNA polymerase. The DNA polymerase
గూగుల్ పే చేయొచ్చండి.
can
గూగుల్ పే
exchange the
చేశాను.
new chain of the DNA in the presence of
Ah.
RNA polymerase.
ఉంది. గూగుల్ పే నంబర్ ఉన్నది.
So the primase enzymes are generated
మాట్లాడతాను.
the small nucleotide of the RNA
Okay.
molecules known as the primers. These
సరే
primers
సరే మీకు దగ్గరకు వచ్చిన తర్వాత
goes to the known the replication
Sure.
for the progress
అదేంటి
towards
అట్లా ఎట్లా వెళ్ళిపోతారు
origin.
మీరు కాలానికి? ఇప్పుడు 3:30
The
అయింది.
elongation is mediated by the DNA
3:30 అట్లా
polymerase
వెళ్తారు మీరు?
enzyme. This enzyme is required for the
అవునండి. మా
free
కాడ నుంచి ఆఫీస్ అట్లా
free
వెళ్ళిపోతారు.
OH of the pre-existing polymer nucleotide for initiation of the DNA replication. So the DNA polymerase
నేను చెప్తానండి. ఆయనకు వచ్చిన తర్వాత
adds
మీకు
the deoxyribose nucleotide for the free OH of this growth
ఓకే.
nucleotide chain. This process of replication results in the formation of
సరే అండి.
daughter DNA.
ఓకే. మీకు ఎన్ని కావాలి? 2 కేజీలా?
Formed DNA and one strand
నాకు
replication
2 కేజీలు అనుకో, 4
strand
కేజీలు
and
అయిపోయినాయి ఇక్కడ.
another one is newly
సునీల్
formed
ఒకటి
DNA.
5 కేజీలు అయిపోయినాయి. ఓకే.
This is
ఇంకెవరు
process
తీసుకొని
known as
5 కేజీలు ఫిక్స్.
conservative DNA replication. So another one is the
మీరు 2 కదా? ఒకటా?
termination. Here
ఓకే సార్.
in E. coli and other prokaryotic organisms, the termination is singly
సార్ ఎక్కువ మందిని అడగటం
held
వేసుకోవచ్చు.
by the specific
సోమరాజుని అడుగుతాను.
called as a tear element. Some enzymes are involved in the DNA replication, that is the DNA polymerases. So these DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3 also, which are involved in the replication of the DNA. And DNA helicase is also involved. So DNA helicase, this enzyme is unwinded and DNA which promote the separation of the two parental strand and establish the replication 4. So DNA garage also DNA primers. These also helped in the DNA replication. Okay. So types of the RNAs. Yeah. Types of the RNAs. The RNA is another important nucleic acid based on the DNA. It is commonly located in the nucleus and cytoplasm. So it is also found in the mitochondria and chloroplast also. The RNA also genetic element in some organisms. So the best example, few plants viruses, plant viruses and some animal viruses also there. The RNA is single strand polynucleic acid. RNA has ribose sugars. So here the adenine and guanine, cytosine, and uracil is also present instead of the thymine. So RNA is a single strand molecule, and hence there is
ఎక్కడున్నారండి?
and pyrimidines
మీ సేవలు మాకు అవసరమేనండి.
honest one. So please keep your mute. Please
మొన్న మన సిల్వర్ నుంచి మీరు డబ్బా తెచ్చారు కదా ఒకటి.
all of you please keep your mute.
ఆ డబ్బాని కొంచెం ఇస్తే మాకు అదే డబ్బులు మీరు
Audio.
ఇస్తాం.
So messenger RNA. So here three types of
అర్జెంట్ ఏం
RNA
లేదు,
are there:
అర్జెంట్ ఏం
messenger
లేదు.
RNA, ribosomal RNA, and transfer RNA. Please
ఎన్నింటికి
keep
వెళ్తారు మీరు
all of you
అక్కడికి
keep
కిందకి?
your hello. Please keep your mic.
5ంటికా? 5ంటికి.
Please mute
ఇప్పుడు
your audio. Mute your audio in mute.
రామయ్య గారి బండి వస్తుంది ఆవిడ
So
మాకా నుంచి.
messenger RNA carries genetic information from
మాకా నుంచి.
mitochondrial RNA to ribosomal
ఆ
of the
మంగళ
protein
మీదకి బెండ్ అని
synthesis.
వెళ్ళింది.
So it is the function as the messenger
ఆ బండిలో
RNA.
ఇచ్చేస్తే ఆవిడ రేపు వెళ్తున్నారు
That's
డ్రైవర్
named as
సిల్వరి.
mRNA. It contains 5 to 10% of the total quantity of the RNA present in the cytoplasm.
అదే ఏదో ఆలోచిస్తున్నా.
Another one is
మాకా
the
నుంచి వచ్చారు, దాని
ribosomal
పైన పెట్టింది
RNA.
రామయ్య గారు మంగళ
So
వచ్చేసరికి.
RNA present in the ribosomes known as the ribosomal RNA or insoluble RNA. It consists of large part of
అదే
the
గంట గంట గంటలకు వస్తారా?
protonuclear RNA. It consists of a single strand nucleotide
సరే.
which is folded in several places
ఓకే.
of pseudo
సరే. ఓకే.
helix. So the ribosome of the prokaryotes cells of 70 types of the ribosomes are there. So another one is the transfer RNA, here tRNA, that we call as the tRNA. So tRNA is also known as the soluble RNA or sRNA or adapter RNA. It is constitutes the about 10 to 15 of the total weight of the RNA, and it is smallest form, and it is composed of around 95 nucleotides. So which is proposed the clover leaf model to explain the molecular structure and activity of the tRNA. Okay, the loops in the form clover leaf here formed due to the folding of the single polynucleotide which on the bone back. So here the clover leaf tRNA model, here amino acid adapter site. The amino acid acceptor site is the 3 end and tRNA. It shows the nucleotide that is the CCA, and that one is TC loop at the 3 TC arm or thymine arm is present. It shows 3 unpaired nucleotides of the mRNA. It has modified base pseudoridine. This is known as a loop or arm. So another one is the D loop or DHU loop. This one is the D means DHU means dihydrouridine. That one. So here the amino acid synthase loop is made up of 8 to 12 unpaired bases are there, which is helps in the identifying the amino acyl synthetase enzyme. So D loop is present between the acceptor site and anticodon loop. So anticodon loop. This loop consists of amino acid sites on the opposite direction. This contains the anticodon and recognized the codon of the mRNA. These are no need to have tRNA equivalent the number of the codon. So along with these loop, there is the one starter arm known as the extra arm. The extra, the exact function of this is we don't know, not known this one. So cloverleaf RNA shows secondary structure depending on the protein synthesis. Amino acid attached itself to 3 terminal of the acceptor system. So all the tRNA contain the large amount of the unusual nucleosides are there. What is the function of the RNA and DNA here. The DNA is the genetic material. It is promotes the synthesis of proteins and regulate the various biochemical reactions of the cell. The RNA takes part in the protein synthesis. mRNA carries genetic inform protein synthesis from the DNA to ribosomes, where the tRNA transfer amino acid to ribosome. Okay, so here, so thank you attending classes here.
Thank you, sir. Sir,
Okay,
can you
thank
give us important questions also?
okay. After that, I will give.
Okay, sure, sir. Thank you.
Excuse me, sir, which